But there's also times you cannot direct get the sequence information. Although now we always say, oh, getting a sequence is relatively easy. But not always possible to get the sequence information. So we are going to talk about some other ways to look at or to estimate the sequence divergence. For example, here, we want to look at an RFP gel, and that's the fragment, like a DNA fragment. Nowadays, we pretty much always use PCR to amplify it, and we usually receive enzyme to cut it. Then after retrieving and digestion, and we can run it on the gel, and look at the fragment differences. Okay. Then we have to know how we can indirectly estimate the substitution rate. So you have to have a little bit every about how the pattern is different from one to the other. So if every fragment is the same, then they were cut by a restriction enzyme. And you have to know how many recognition sites for that particular enzyme. Some enzyme recognizes 6 nucleotides. Some enzyme only recognize 4 nucleotides. Some enzyme recognize 8 nucleotides or even more. And if a site which cut or uncut, so if we look at the sites between, for example, the last one. Okay, so that last one I draw is a little bit higher, if higher in the molecular weight. And we compare that to the third to the last. It cut to one site, assuming they have a homology. And so this sample cut by one restriction enzyme, and assuming they recognize 6 nucleotides, and the other sample didn't cut. So you know there's a differences at the recognition site. So that's why I say you have to know a little bit more about which enzyme is used in this experiment. So assuming that's an equal R1 site, and there are it's a 6 cutter, you recognize 6 sites, and the minimal change for these 2 samples is there's 1 nucleotide differences at that recognition site between the 2 sequences. That's a minimal change. So then you can say, okay, between the 2, at the least, there's 1 out of 6 changes at that position. You only sample 6 nucleotides and you observe 1 change. You can use multiple enzymes to do the same thing, and you can analyze a little bit about the recognition site property, how likely they overlap or non-overlap. If you choose all the non-overlap restriction site, you can still estimate the number of changes between the 2 sequences. Actually, that's what people earlier used the mitochondrial IFLP. They can also estimate the divergence time and also estimate the divergence time between the 2 mitochondrial genome. So this is an indirect method, but you have to give additional information to look at that. In amino acids, sometimes you only have amino acid if you are working on proteomics. So protein is the one we think, although we know a lot of RNA is very important too, but we still focus on a lot of the time on the proteins. And now with the high throughput method, you can estimate the protein sequence. Okay, you can collect a lot of protein sequence at one time and look at the protein sequences difference between 2 species. Then the question is, without DNA sequence, How could you do that? And it's very simple because early days in molecular evolution, there's only protein sequence. Before we know how to sequence DNA in late 70s. And not nowadays, like we have tons of DNA sequence available for all the analysis. But this is relatively simple just by counting the differences. So on this slide, I try to show you how to compare the two amino acid sequence, do alignment, count the differences, and we can calculate the proportion, the same thing, the observed proportion of differences in or between the two sequences we aligned. Then we can calculate the distance. Just try to take the minus natural log to that sequences.